All right, welcome everybody to Ann Arbor's American Statistical Association chapter meeting. We are very lucky today to have a great room and a great speaker, Brady Ryan. He's a PhD student in the Department of Biostatistics at the University of Michigan. And his dissertation focuses on two main areas of statistical genetics: developing and applying methods for genome-wide association study, summary statistics, and developing methods to identify causal variants in molecular pathways for human traits and disease. He's developed methods in the areas of rare variant aggregation testing, GWAS meta-analysis, and Mendelian randomization. And he also collaborates with the metabolic syndrome in men, the METSIM study. All right. So please join me in thanking Brady, soon to be Dr. Ryan, I'm sure, for coming to talk to us about FinemappingMR. Yeah, go ahead.
Well, thanks everyone for coming here. Today I'm going to talk about my project, FinemappingMR, a novel Mendelian randomization method using statistical fine mapping. So I'm going to start with a brief introduction to statistical genetics. I know some people here don't have a really extensive background in StatGen, so I'll start with that. I'll give an introduction to Mendelian randomization before introducing our method. I'll talk about some simulation and some applied results, and then talk about some conclusions and potential future work. So we're going to go all the way back to high school biology at the beginning of this talk. The human genome is made up of long-stranded DNA, which are essentially just a sequence of letters, which are either A, C, T, or G, and these are packaged into structures called chromosomes. So all of us have 22 pairs of autosomes, one pair of sex chromosomes, and this is about 3.3 billion base pairs long. So you get one chromosome from the mother and then one from the father. So if you multiply that by 2, you actually get 6.6 billion base pairs. And this here on the left, on the right, is just a little diagram of what DNA kind of looks like with the helix. And here's the central dogma of molecular biology. So it says that genetic information flows only in one direction. Here we have a typical gene region. So our exons are what contain the protein coding information. These intronic regions do not contain any protein coding regions. This promoter region kind of turns on and off the gene. This DNA is then transcribed into RNA, which is then spliced to get messenger RNA that's then translated into an amino acid sequence before being folded and then having some post-translational modifications to become proteins that circulate in our bodies and do various types of things. So some quick terminology. A single nucleotide variant is just a single nucleotide in the genome which is altered. Sometimes we'll call these SNPs if it's changed in greater than 1% of the population. A locus is just going to refer to a specific location in the genome. This could either be at a single nucleotide or could be multiple nucleotides together. An allele is the particular nucleotide at a locus where a single nucleotide variant exists. So in this little figure, I have this purple individual has the A allele at that SNP that you can see. The blue individual has the G allele, and then the orange individual has the T allele. And so variants can be more complex than this, but throughout the talk, I'll just use the term variant to refer to a single nucleotide variant or a single nucleotide polymorphism of SNP. So all three of those things kind of mean the same thing for this talk. And kind of at the crux of what we do in statistical genetics is genome-wide association studies. So in a GWAS, we're interested in identifying variants in the genome that are associated with a human trait or disease. So just thinking about a disease for a second, you can enroll a set of cases and a set of controls. You can obtain genotypes at every position in the human genome for those individuals. And then at a high level, you can just look at the variants that have higher frequency in cases than in controls. You can perform a statistical test and then represent this information in a Manhattan. So here on the x-axis, you have the chromosome. It's kind of hard to see the labels, but each color represents a different chromosome. And on the y-axis, the negative log 10 p-value from performing these GWAS. And so we do a ton of tests. And so in general, we declare something to be statistically significant if it passes the von Ferroni threshold of 5 times 10 to the negative 8. And this dashed line, which is kind of hard to see, indicates a genome-wide significance threshold, and you can see a couple loci along this genome that have genome-wide significant hits. And this is kind of the pattern that we expect from a Manhattan plot, where you don't just get a single genetic variant reach genome-wide significance, you'll have multiple in the region that do. And I'll talk about that in a little bit. And the one other thing I want to talk about for GWAS is that they're kind of a special case of association study, whereas generally in statistics, we can't assign causality when we identify association. But in GWAS, say for some certain scenarios where there could be population stratification or sort of mating, generally associations that we can find can be interpreted to be causal, because there aren't really anything that you encounter in your daily life that could actually affect your genotypes. And so it's kind of free of confounding that's generally associated with typical statistical analyses.
Are you sharing your screen?
I am not.
I'm not, because that might be hard for anybody on the video.
It may be difficult.
Yeah.
Thank you for that.
Yeah, good call.
Thanks. I got another question.
Of course.
So in these variants, the locus is the same for all, even though where you're studying it, it's one locus, and whatever the multiplicity of phenotypes or gene or whatever it is that can go there, right? And you're looking across the loci on the x-axis, is that right?
Right, that's across, yeah, all the loci in your genome. This would be for a single trace, and then just comparing. Like at a high level, you can consider just like a 2-sample t-test between your cases and controls. Of course, in practice, we do a little bit more complicated than that. There are some confounders that you want to adjust for, but yeah, it's across the loci in the genome is what's on the
Because I don't have much exposure. It sounds like there's a lot of locations in which variants occur in the genetic population. That's the kind of sense I don't really.
Yeah, there's millions of genetic
see.
variants, hundreds of millions if you include rare variants. So yeah, that's why you're doing a ton of testing when you do these GWAS.
Yeah, thanks.
So
Yeah.
I don't want to slow you like
No, go ahead.
slide 7 with questions.
Go ahead.
I think it's interesting, this idea that you don't really have confounding.
Mhm.
I just wanted to test that theory with this question.
Sure.
You've got a couple genes there that have gene cases and control.
Yeah.
Right. So what if only one of those genes is really causal and the other is just some kind of correlate?
Yeah, so I'm going to talk about that in a little bit. Yeah. So that is one of the. So yeah, I think in 2 slides I'll start talking about that. But first of all, genome-wide association studies have been kind of wildly successful in identifying associations with human traits and disease. So this kind of crammed picture, each of these little rectangles that you may or may not be able to see on the left is a chromosome. And then there's lines coming out of the chromosome which indicate locations on that chromosome where there's been a genetic association identified. And then all of these colors correspond to different classes of phenotypes. So almost everywhere across the genome we found significant associations with some trait or disease.
Is this the rainbow of humanity?
Yeah, there is some legend that goes along with it to indicate the different colors, but there's too many of them to put on a slide. And so the issues in interpreting GWAS signals, the number one issue is that due to linkage disequilibrium, which is essentially correlation between variants that I'll talk about in a little bit, it is difficult to identify what the true causal variants are. And this is why you get these typical GWAS peaks that we just saw in that Manhattan plot. And I'll discuss this a little bit more in the next coming slides. And then the other problem is that the biological pathways through which these causal variants act to impact disease risk is not always clear. So I'm going to start with LD, and LD is a phenomenon that occurs due to recombination that occurs during meiosis. So we all get one chromosome from the mother, one from the father, and then we pass those on down to our children. We don't just go across them and then pick at each genetic variant randomly which one to give to the offspring. What actually happens is that these 2 chromosomes line up and they exchange genetic information in what's called crossing over or recombination. And then eventually we will pass one of these 4 onto our offspring. And so you can imagine that because these are kind of inherited in chunks, that if two genetic variants are physically close together, they're more likely to be combined together, which means they become correlated, and then we say that they're in LD with each other. And this complicates the assignment of causality to any single variant. So here on the left, you have like a typical LD block for just, I think, 8 variants. These are just kind of supposed to be color-coded by R squared, and then you can see that these 4 variants very close together and are really highly correlated with each other. And that leads to this kind of typical GWAS peak that you can see on the right, where again on the x-axis, you have the genomic loci, the GWAS signal with the negative log 10 p-value on the y-axis. And what can happen is due to the correlation structure, the most significant variant, this one here, may not actually be the causal variant.
Oh.
So statistical fine mapping is one way to help resolve these issues, and it's something that we're going to incorporate into our Mendelian randomization model that I'll talk about later in this presentation. So as I just mentioned, due to LD, the lead variant in a region may not be the causal variant, and there may also be multiple causal variants existing in the same region. So here on the right, I have another little toy example where I have 1,000 SNPs on the x-axis, and on the y-axis, again, the negative log 10 p-value. This yellow variant is the strongest marginal association, so it's the lead variant in the region, but the data that actually generated this was SNP1 and SNP2 being causal, and those are indicated in red. So you kind of, yes.
I didn't want
No,
to
go ahead.
finish your sentence.
Yeah.
Okay. So we're talking about this disequilibrium. Is there a way of thinking about what things would look like in equilibrium? Is it linkage equilibrium? Is that when they're all mixed?
Yeah, linkage equilibrium would be basically if everything was uncorrelated.
Okay.
Which
No physical association has any impact or
Right.
location and regionality, all that other stuff. It's just a big gray map. Okay, got it.
yeah, yeah. And so we apply these fine mapping methods in order to try to disentangle these LD patterns to find what the true causal variants actually are. So one such method is SuSiE, and this was actually taken from the SuSiE paper. And this bottom plot is the result of running SuSiE on this top region. And here, the bottom region, instead of a log 10 p-value on the y-axis, I have a posterior inclusion probability, which is essentially the probability that a variant is causal, given that there is a signal in the region. So we present these in terms of these pips as well as credible sets. And so you can think of a credible set as being an independent signal that exists in the region, and then the variants within those credible sets as being plausibly the variants that could be contributing to that signal. So when you run SuSiE on this region, you do indeed identify 2 credible sets, the one circled in blue, and then it says the causal variant corresponding to that signal could be any of the 3 variants circled in blue. The green circled variants are a separate credible set, and again, any of the variants in there could in theory be the causal variant. And then we've excluded the yellow variants, and so we're able to say that although it's the strongest marginal association, it's not actually the causal variant that's leading to this plot that we have at the top. And so the other issue interpreting GWAS signals that I'm going to talk more about today is that these biological pathways through which they act is not always clear. So actually over 90% of GWAS signals are non-coding regions of the genome. Actually only about 2% of the genome even codes for proteins. And so the majority of the GWAS signals that we get are actually not in these coding regions. And when they're not in these regions, they don't always act with the nearest gene. And even if they're in the regulatory region for a gene, these regulatory regions may actually overlap or be in LD with each other. And
Is the coding a direct expression rather than latency that I've heard about when you're talking about it codes?
Yeah, so coding I would refer to as, it contains genetic information that gets transcribed into RNA that then gets translated into protein.
Rather than waiting to be switched on later.
Right, so the regulatory regions kind of do the switching on and off, and that's where actually the majority of the GWAS signals that you find are.
Oh,
So they're actually in a kind of promoter or enhancer regions generally.
thanks.
And so one way to try to do this is to use Mendelian randomization. And I'm going to talk about a specific type of MR called drug target MR, where we're interested in identifying proteins that are potentially causal for disease. So I'll pause quickly to take any questions. Otherwise, I'll move on to an introduction to MR. Okay. So of course, everyone here in this audience knows that correlation is not equal to causation. So if we're interested in, for example, studying if vitamin E has a causal effect on heart disease, you can think of all these confounders that kind of make this difficult to assign causality to. And one way to test a causal effect is to perform a randomized controlled trial. This is, people here probably know more about randomized controlled trials than I do, but they're kind of the gold standard for testing the causal effect of an exposure on an outcome. Of course, they have their own issues that I won't get into, but essentially what it is that we would enroll a set of individuals. In this case, I have these blue bowling pins, which are diseased individuals, which I've enrolled for my study. They're randomized into a treatment and a control group. The treatment gets some kind of intervention. The control group will get some comparison or standard of care. And then after some follow-up period, we can just compare the proportion of cured individuals in the intervention group to the comparison group. You can kind of formalize this by looking at this directed acyclic graph. But really, in theory, randomization should be your only cause of treatment. You can imagine that there could be some confounders that are at least associated with being in the treatment or control group as well as with your outcome. But the good part is that randomization, in theory, balances these confounders, allowing us to actually estimate a causal effect gamma of the treatment on the outcome. And what Mendelian randomization does is it attempts to replace this randomization variable with genetic variants in an observational study. So there's some assumptions that go along with MR that essentially make this directed acyclic graph look like the one I showed you on the previous slide. The first is the relevance assumption. So we need to choose genetic variants that are strongly associated with the exposure. This is generally easily done because we just perform these genome-wide association studies on the exposure, and we can just choose variants that have a really low p-value, for example. The second is the exclusion restriction, which says that if your genetic variant is causal for the outcome, it's only causal through the exposure. And so in general, this is probably never true for Mendelian randomization analysis. And so you need to do things to try to account for this. And in our model, we'll explicitly model some horizontal pleiotropy term, which I'll talk about when I introduce the model. And the third is the exchangeability assumption, which I kind of already discussed, that there's basically no confounding of your genetic variant of the outcome. And there are ways that this can happen, such as population stratification, but these are generally things that are very easy to account for. And so we generally just assume that this is kind of true based on kind of being randomly assigned alleles at birth.
So I have one question. So
Yeah.
here the confounders are measured or unmeasured?
I mean, in general, unmeasured, I would say.
But if the confounders were measured, some were measured, some were unmeasured.
Sure.
With them, the measured ones can have association with the genetic variant, right?
Yeah, in general, no. Like you wouldn't expect that there's any kind of confounder that you can think of that could actually affect the genotypes that you have.
Okay.
So they're kind of conceived, you get them at birth, and then they don't change over your lifetime.
So this
So
is like more like a special type of instrumental variant.
it's exactly a special type of instrumental variable analysis. So you can measure the effect of your genetic variant Z on X, your exposure with beta ZX. You can get this from a genome-wide association study. You can do the same thing for the genetic variant of the outcome, beta ZY. And then if all three of those assumptions are satisfied, then this true causal effect gamma is equal to the ratio of beta ZY and beta ZX. And so if you just had a single genetic variant, this is a way to get a really simple Mendelian randomization estimate. And as you mentioned, we also refer to these instrumental variables or IVs. So later I may say the words IVs or instrumental variables. These are just the genetic variants that I'm talking about that we've chosen for our MR analysis. And the particular area that we're interested in is using MR for drug target prioritization. There's a growing interest in doing this, where essentially we're using molecular traits such as protein expression. So the amount of a given protein that's circulating in a tissue at any given time, as an exposure in our Mendelian randomization analysis. So typically when you take some drug, it targets some protein. And so you can kind of think of having genetic variants that reduce or increase this protein as a proxy for a drug target. We often only use the cis region of the exposure. This just means basically the protein coding region, plus or minus some number of base pairs. And there are two issues with this. One, there's the potential for horizontal pleiotropy, which is a violation of our exclusion restriction, which I'll talk about. And also that traditional MR methods use only independent variants, which makes them underpowered when using only a cis region where there's a lot of correlation, as I showed earlier. So the first issue is horizontal pleiotropy, and this can kind of happen in two different ways. Here on the left is a case where you've chosen a genetic variant C to be an instrument in your analysis, and it's actually causal for the outcome through some other pathway. This is just like a direct violation of your assumption. But in practice, what's more likely to happen is this case B, where you've chosen a variant Z1 that might actually be a valid instrument, but is correlated with a second variant Z2 that's causal through another pathway. And so both of these will cause bias in your causal estimate, and this could be in either direction. While we might think that this could be an issue in when we use molecular traits, as I've already mentioned, that the regulatory regions of these genes may overlap or be in LD, and so variants that are close together, even if they're in a single gene, could still be affecting other genes outside of through X. The second issue is that traditional MR methods use only independent SNPs, and for molecular traits, it's likely that they have multiple causal variants in a single region. So here on the left, I have an example of a Manhattan plot in the cis region of the NCAN protein, which is a protein I'll talk about later. You can see that there's just a ton of variants that pass our genome-wide significance threshold, which is my red dashed line. And so, as I said before, you can run SuSiE on this region to identify what are the potential causal variants. So this is what I've given here on the right. So now instead of the log tan p-value, I have the posterior inclusion probability. And we can look through this and look at how many credible sets were actually identified. So our first credible set is a single variant with a posterior inclusion probability of essentially 1. It's very likely a causal variant. There's another credible set with a posterior inclusion probability of 1, very likely to be a causal variant. And as we go on, we have another credible set, 4 variants, one of them likely to be causal, a fourth credible set with 2 variants, and then a fifth one with 3 variants. And so putting all this together, basically SuSiE thinks that in this 1 megabase region, so there's a million base pairs, in the protein coding region for NCAN, there's likely at least 5 causal variants. And the usual procedure for traditional MR methods is to just take all variants that have a p-value less than 5 times 10 to the negative 8 with the exposure, and then a pairwise R-squared less than 0.01. As I've mentioned, multiple causal signals could be in LD with each other. So in this little toy example, I just have 7 variants, and I've shown that the 3 red shaded blocks are causal SNPs, so there's 3 causal SNPs in the region. The first thing you would do is to perform a genome-wide association study on these 7 variants, and then I have the dashed line representing genome-wide significance. The first step, you would look at which variants reach your genome-wide significance. In this case, it would be all of them. The next step would be then to look at the correlation structure, and you can see SNPs 1 through 4 are highly correlated, SNPs 5 through 7 are highly correlated. And so a traditional MR method would only take the top SNP from each of those 2 LD blocks, and so we'd just use SNP 2 and SNP 6. But of course, I showed you that there's 3 causal SNPs, and so the idea is that we can incorporate multiple correlated IVs. This has the potential to improve the power. So finally, the goal of this project was to basically propose an MR method that can use multiple potentially correlated causal variants while also taking into account horizontal pleiotropy. And we're going to do that by incorporating the SuSiE fine mapping model, which I've kind of heard some results from already this time. So that was an introduction to the project. If there's any more questions, I'm happy to take them. Otherwise, I'll move on to talk about the method.
Just a thought, actually, and I can't claim to be an expert in this area.
Yeah.
There's something in machine learning called embeddings that has recently come to my attention. Have you heard about these?
I've heard the word embedding in my life, but I could not tell you at all what it is.
Yeah, so these large language models where they put words that mean similar things together.
Right.
And it's very much related to this idea of the core, what's correlated in this big mess of data and what's not. And that representation of the correlation, I think.
Yeah, so I know that there are like machine learning models that kind of just look at protein DNA sequence and take a bunch of features like conservation of variants for other species, all these different effects, and then try to predict variants might be causal based on those things. And I guess it also must look at the correlation. How that could be used, I'm not.
No, this whole idea of representing the, how similar, you take 2 things, how similar are they, and how different are they sort of measured?
I see.
And then there's some kind of propagation of, it's different from principal component analysis
Uh-huh.
for reducing data dimension, but
I see. Yeah.
I think it might, I'm just getting interested in it myself, but
Yeah, there might be a way to
the
like
statement
choose.
of the problem reminds me that there's,
Right, there might be some way to choose variants that doesn't do this kind of the.
yeah, of course,
I see.
this is not.
Yeah, yeah.
This is not, there might be stuff to learn still.
Oh, yeah. I'm sure there is. Okay, so I just briefly have some slides on the method. I'm really not going to go too in depth on the method, but you can kind of sum it up by looking at this directed acyclic graph. This is like exactly the one that I showed you for the MR assumptions, except that now we actually allow violations of this horizontal pleiotropy assumption by directly modeling a direct effect of G into Y, which is alpha. Another thing that's different about this directed acyclic graph and our method is that we don't actually do any variant selection like I showed you previously. We input the entire cis region, and essentially we're going to more or less run SuSiE to kind of get some automatic variant selection as we fit the model. But this G here now is a matrix, not a single genetic variant like I showed you in previous slides. So this B is a vector and this alpha is a vector. And we're interested in estimating gamma. So in terms of structural equations, you can write them as x equal gxb plus epsilon, y equal gamma x plus gy alpha plus epsilon y. Here I've written it out as if the x and y were measured in non-overlapping studies. That is in general the best thing for Mendelian randomization because it kind of removes any confounding of x and y, environmental confounders. But you can adapt this to work in a one sample or a scenario where there's some subset that overlap. If you plug 1 into 2, then you get this new equation for y. And so basically we use this new equation for y and our equation 1 for x to form a Bayesian hierarchical model where we place the SuSiE priors on B and alpha. So all of that SuSiE that I showed you before, it comes from placing the prior on B and alpha, so it's sparse. So we assume that most of the variants in the region don't have an effect, but that we do allow for multiple causal variants in the region. And also we're modeling the correlation between them by using these priors. Two of the epsilon terms you just assume are normally distributed, and we place a normal prior on gamma with mean 0 and variance sigma gamma squared. If you want to use something approximately flat, then you can choose this to be, the variance should be something large. So all I'll say about the posterior estimation is that we use variational inference. Kind of sampling techniques are impossible in this case. The parameters are, there's 10 times the number of variants that you have in the region. That's the number of parameters that exist. So it's kind of impossible to do anything other than variational inference. For those that aren't familiar, you're essentially trying to find a distribution that's close to the posterior P in terms of KL divergence. But I have some slides that I can show later, but for now I won't really talk about that too much.
So I have one question.
Sure.
So this is definitely with how many genes are there, kind of like G matrix, like.
Right. So in this case, we're going to talk about just a single, like a cis region. So like in general, you would just have a 1 megabase region, and then however many variants are in that region generally ranges from like 3 to 6,000. You can extend this to have multiple independent genetic regions, and we've done that, but it's kind of computationally inefficient at the current time.
So I was thinking if I take a polygenic risk or something like that, instead of taking all the genes together like this. So have you compared your method with something like that? I know it's like an approximation with that.
Yeah, so PRS is kind of like, it's kind of like MR, but you're like you just don't do the same stringent variable selection that you do in MR, right? Yeah, I think in general, and so for this particular scenario, when you're just looking at a cis region, I have a feeling that there's probably quite a bit of false positives that you would get from the horizontal pleiotropy, because you generally don't model that when you're using a PRS. I'll show again some just traditional MR methods that use just a few variants, and then they have literally severely inflated type 1 error when there's horizontal pleiotropy. So I think you kind of increase your risk if you just use more variants. So that's what I would assume. Yeah. But it has like a similar, it's kind of like a PRS, like we're still using it's kind of like using all the variants in the region, just like a small PRS, and there's shrinkage on the the X is through this SuSiE prior.
Wow.
Okay, so I'll talk about some simulation results first. We simulated a cis region with horizontal pleiotropy. I used GWAS Brewer. This is an R package produced by one of my supervisors, Gene, to simulate individual level GWAS data. I used an LD block that had 863 SNPs from chromosome 19 and 1,000 genomes, which is a publicly available resource with individual level genetic data on 2,500 individuals. You can think of lining up these variants from 1 to 863. And then the first step would be to pick 5 random causal SNPs for your exposure and 2 random causal SNPs for your outcome. So these SNPs that I've highlighted in blue will be causal for your outcome, but not mediated by X. And then these 5 causal SNPs for X will be causal for Y when there's a direct effect of X on Y. And I'll show that in a directed acyclic graph in the next slide, so it might be easier to wrap your head around. But then you assign an expected heritability to each causal X SNP. So a heritability is the proportion of the variance, or the proportion of the variation in the exposure that a SNP explains. So we set this to be 0.01, and we're using standardized exposure. So this means that each of these variants explains 1% of the variation in the exposure. So these are really large effect sizes, but actually not that uncommon if you're looking at, for example, a loss of function variant for a protein in the cis region. When there's a causal effect of X on Y, these variants will also affect Y, and then their expected heritability will be gamma squared times 0.01. We picked gamma values that basically correspond to the exposure explaining between 0 and 16% of the variation in the outcome. And then you need to assign an expected heritability to each causal Y SNP. We set it to be half what it was for the exposure. Still, this is a very large effect size. And essentially what you do is you just draw effect size, a normal distribution with mean equal to 0 and variance equal to heritability, and then you kind of generate from a linear model. We simulated 5,000 individuals for the exposure and outcome with no overlap. Repeated this 1,000 times with different causal SNPs each time. So even though I showed these 7, each iteration is a different sampling of causal SNPs. So you want to look at this kind of in a directed acyclic graph. You'll have these 5 causal variants that directly affect your exposure. You expect them to explain 1% of the variation. When there's a causal effect on your outcome, then each of these also explain gamma squared times 0.01 of the variation of the outcome. And then you always have these 2 other SNPs that only affect your outcome. They do not affect your exposure. And an important note is that all 7 of these variants could be correlated. So we compared against traditional MR methods. I kind of described LD pruning earlier. This time we used a more lenient threshold of p less than 10 to the -6 and a pairwise R squared less than 0.01. We use 5 different methods. This is a table that kind of summarizes them. The first column here indicates the number of instrumental variables you need in order to get an estimate. There's one other method, RAPS, which accounts for pleiotropy, and there's one other method that accounts for LD, which is GSMR. So for GSMR, since we can use correlated variants, we can't really do this LD pruning in the same way. I basically run SuSiE on the exposure region, get some credible sets, and then you pick one variant from each credible set, and you just don't worry about the correlation because you can input the correlation when you're getting an estimate from GSMR. So this is a big table of results. So I'm going to go through these individually to kind of show you what I want you to see. But each column is a different method. Our method starting here on the left. And as you move down the table, you have increasing gamma from 0 to 0.4. And then the cells indicate the mean gamma estimate across the iterations. And in brackets, I have the standard deviation. Before going any further, I just want to make a note of this on the bottom. Each of these methods requires a different number of instrumental variables to run. If I go back, median requires at least 3, IBW at least 2. So although I picked 5 causal variants, when you actually sample the effect sizes, run the GWAS, they may not reach a p-value of 10 to the -6, or they may be correlated with another instrument, so they're thrown out in the LD pruning part. So not every iteration is going to have at least 3 IVs, and so that's indicated here on the bottom by the differing number of iterations that we could run each method on. Okay, so the first thing to note, our method has lower standard deviation across the iterations than SMR, GSMR, and RAPS, and is also a little bit more accurate, especially as gamma increases. If you just compare to IVW and median, it looks like we have higher standard deviation, especially compared to median, and at least at gamma equal to 0.4 for IVW. But as I already mentioned, these methods require more instrumental variables in order to run. So they can only run in situations where they inherently have more information. And so you would expect less variability in those results. So to give a fair playing field, what you can do is just restrict to the iterations that had at least 3 instrumental variables identified. And you can see when you do this, that FinemappingMR is accurate under all scenarios, more accurate than the other methods. And it also has a lower standard deviation across the estimates, across the iteration. So we're mostly going to be interested in power and type 1 error. So that's what I have in this table. Again, 1 column for each of the methods. The first row will be your type 1 error scenario. As you move down the table will be power scenarios. If you look at the type 1 error, in this case, you just look at across all the iterations, there's no method that controls type 1 error. We are able to obtain better control than median RAPS, SMR, and GSMR, but I will show in a little bit that under the specific cases where we can actually control type 1 error. And it has to do with, of course, the correlation structure. But if you look at power, and it's, none of the methods can really control type 1 error, so it's hard to give a fair power assessment. But we both had better control in general of type 1 error, but at the same time are also able to have more powerful results than the other methods. You can also look at coverage. In general, we kind of get better coverage as we increase gamma. At 0 and 0.1, we're kind of outside the 0.95 coverage. The other methods kind of have varying different coverage rates. IVW tends to get a little worse. SMR and GSMR are kind of always bad. RAPS kind of goes up and down.
How long did your simulations take to run?
So these ones are really fast. They don't take very long at all.
Great.
I would say in general, it doesn't take long to run this method on just a single genetic region. So if you increase the number of regions, the problem is that you have to read in the LD matrix, the correlation matrix for each of the regions. Mm-hmm. So you get above like 5, then you're having potentially like 4,000 by 4,000 matrices, and you have 5, 6, 7, 8 of them. It's just, and then you need to do this huge for loop in order to actually estimate the posterior that can be parallelized. So it can get quite slow if you increase the number of regions.
Sparsity helps, but
yeah. So I don't think that's not really the case that we'd really recommend applying the method, because if you have a lot of instruments, actually IVW or RAS will actually give you a really good result anyways. The really benefit is in these cases where you have a small region with not a lot of instrumental variables to work.
That's important to notice.
Yeah. But in the cis region, you can see that especially SMR, GSMR, and RAPS, in QQ plot, this is the null scenario, have really kind of severely inflated systematic inflation of the type 1 error. Median IVW and FinemappingMR kind of squished down there, but a little bit of inflation, but it's not too bad. But one thing you can do to kind of parse why we're getting these type 1 errors is by looking at the maximum observed R squared between the 5 causal X SNPs and the 2 causal Y SNPs. So again, have 863 variants, choose 5 to be causal for X, 2 to be causal for Y. You can then calculate the correlation between, you can start with 862, calculate the R squared between 862 and 7, 862 and 863, do the same thing for 5 and 7, 5, 863, and then just note the maximum correlation that you get. And then what you can do is to stratify the QQ plot that I showed you earlier by the bin of the R squared. So here on the left, I have the FinemappingMR QQ plot. On the right, I have the RAPS QQ plot. And you can see for us that when you get to basically up until the correlation is less than 0.3, you're still able to not have any of the systematic inflation. Even between 0.3 and 0.5 is not horrible. When you get above that, there becomes an identifiability issue where you can either decide basically that there's pleiotropy and then say that there's a causal effect, or you could say that there's no causal effect and say that there's pleiotropy. So it gets really hard to distinguish those scenarios when the correlation between the causal variants is really, really high. RAPS, you can see, kind of only able to control the type 1 error or not have this inflation in the QQ plot when the R squared is less than 0.1. So
when you just quickly, is there a reason you picked 5 and 2 for the way you?
No, there's not really a reason.
Your intuition helped you with select that, or it's just useful and it worked out. I mean,
we thought 5 is not like unrealistic, but we did kind of just randomly choose it. We are though currently doing updated simulations where we actually ran SuSiE across all the proteins in UK Biobank. And then we have a so we have the SuSiE estimated posterior, so you can identify that how many credible sets do I have. And then so for those simulations, what we're going to do, choose the causal variance from those credible sets. And then you can actually sample the effect size from the SuSiE posterior as well as a realistic posterior effect distribution. And so that's what we're working on right now. You kind of see, actually in those cases, our method generally performs much better than the others. But this is kind of maybe a more fair scenario to them, because the effect sizes just aren't as large, I think. So for the real data application, we wanted to study the causal effect of circulating plasma protein levels on cholesterol in UK Biobank. So circulating plasma protein levels, it's blood is the tissue that we're interested in, and you can measure for each protein that's expressed in your blood, how much of that is circulating at a given time. And we want to study the effect that has on total cholesterol. So we did this for 1,612 proteins. The summary statistics were generated from the UK Biobank Pharmacoproteomics project, and they were generated on 34,557 Europeans. Total cholesterol, we took summary statistics from the Neil lab. This was generated on 344,278 Europeans. This is a common case where a GWAS trait like total cholesterol will have a much higher sample size than a molecular trait like protein expression, because it's just much harder to measure protein expression at a large scale due to cost. So we perform just 3 methods right now, so I don't have RAPS or any of those other methods. This is just because they require LD pruning, and it takes a much longer time, and they just haven't gotten around to doing it. So just comparing against SMR and GSMR. Putting all this together, and then just looking at a Venn diagram of the number of significant proteins at FDR less than 0.05 that we found, our method finds a significantly smaller number of causal proteins. So we just find like 65, whereas the other methods are well over 100. This is actually kind of consistent with the real data with the simulations, because we had like about half the type 1 error rate that they did. And so I just want to talk about one specific example where I think that FinemappingMR had the correct result. It's this NCAN protein that I showed the regional plot for earlier. On the left, I have a table with the SMR, GSMR, and FinemappingMR estimates, as well as their log 10 p-values. And you can see SMR and GSMR finds this really highly significant positive association, whereas FinemappingMR finds basically nothing. On the right, I have an MR plot. So basically in Mendelian randomization, what you're doing is regressing your beta hat y's on your beta hat x's, but forcing that regression to go through the intercept. So as a diagnostic tool, you can make these MR plots, where on the y-axis I have the cholesterol beta, so the beta hat y, and on the x-axis the beta hat x, the NCAN beta. And here I've included every variant that appeared in a credible set when I ran SuSiE. So I can also kind of superimpose the actual estimates that we got. So SMR uses only one variant ever, no matter how many variants are there. It uses only the strongest association with exposure. So that estimate is going to go straight through that line, straight through that point that it used, which it finds to be very significant. GSMR is kind of using one variant from each of these credible sets, and so it's going to try to fit a regression to these 5 variants that it chose. It's kind of, as you can see also, like these 3 credible sets indicate a negative effect, right, whereas these 2 indicate a positive, but these are 2 of the really most significant ones. So these 3 extra ones do kind of bring this slope down a little bit, but it's not enough to bring it back to non-significance because the other effects are just so strong. And then of course, FinemappingMR, you can't see the line because it's basically 0. And I'm going to talk about why I think that is in a little bit, but first I want to convince you that it's probably not the causal protein. So the gene that codes for NCAN is upstream of a potentially causal gene called TM6SF2. This is a genome tract, so it's a bit noisy, but you can see this kind of blue line here, and you can see the label, this is NCAN, and just downstream of it, you have TM6SF2. It's only 12 kilobases away, which is very close. If you just think of, especially the whole genome is like 3.3 billion base pairs long, being 12,000 base pairs away is extremely close, and they can be very correlated in that short range. There's really no evidence to suggest that NCAN is a causal protein for cholesterol. It's actually primarily expressed in the brain, so it doesn't actually even appear in blood very often, and forms part of an extracellular matrix in the brain and spinal cord. And there's really no papers or anything to suggest that it might actually affect cholesterol. However, there are multiple papers showing that the TM6SF2 gene might code for a protein which regulates cholesterol metabolism. This is just a single paper where they showed that hepatic TM6SF2 or liver TM6SF2 regulated cholesterol metabolism in mice, and there are also more studies that show this in humans. So why did FinemappingMR find a non-significant result while the other ones were significantly positive? So if you remember, we also modeled the pleiotropy, where these other two methods did not. So again, we're interested in studying if NCAN is causal for cholesterol. You can kind of put the genes that code for these proteins, the code for TM6SF2 and NCAN below it. We identified two variants, of these two that indicate a positive effect. These both contain one variant, and they were both located in the NCAN protein. So basically, our method is saying we think that these two variants have a direct effect on NCAN. Separately, we identify these pleiotropic credible sets. So we identified one that had two variants. And so what we're saying by saying that we have this pleiotropic credible set is that we think one of these two variants has a direct effect on cholesterol that's not mediated by NCAN. And then if you look at the correlation between these two variants that I've denoted in brown, and then these two that have a direct effect on NCAN, you can see that they're actually really highly correlated. So these variants that have a direct effect on NCAN are actually correlated with these variants that have a direct effect on cholesterol. This is kind of a classic case of horizontal pleiotropy. So FinemappingMR is actually able to account for these pleiotropic variants that are in LD with the NCAN instruments. Just to take this one step further, what you can actually try to do, and I'm not sure this is like the most principled thing to do, but it has a nice result in this case, is that you can basically take these variants that we use for GSMR, and you can regress out the effects of these two pleiotropic variants. And when you do that and plot them over here to get a conditional cholesterol beta, you can see that once you regress out the pleiotropic variants, it just completely brings the cholesterol beta basically to zero. And if you run GSMR then on these 5 variants instead, you actually see that it's non-significant, or it's nominally significant, but of course, due to multiple testing, it definitely won't be significant. So just some quick conclusions. Yeah, of course.
Does that mean that some people have made claims that are going to be undercut by this test? I mean, that it's going to reveal some.
Yeah, I think people are kind of aware of the issue of horizontal pleiotropy. I think like good papers will kind of acknowledge that, you know, even though this is like a causal effect, this could be contaminated. And there's different things you can do to try to look at this in traditional methods. So like if there's horizontal pleiotropy, you would, well, if there's no horizontal pleiotropy and you have, like, you follow your assumptions perfectly, each variant that you use for MR should actually give you the exact same gamma estimate. Obviously, that's not true in practice. And if it's really not true, if it's a variant that's really outlying, then that kind of gives you some evidence that there could be pleiotropy associated with that variant. So people do these things and then try to kind of disentangle what's happening. But yeah, in general, you know, that doesn't always happen. And you know, there could be, obviously, there are definitely papers that, you know, claim these causal effects, but they're just contaminated by nearby genes.
They need to run this test.
Yeah. Yeah. Yeah, just to conclude, we are able to control type 1 error under moderate correlations between pleiotropic variants and true instruments. We found a smaller number of significant proteins in our real data analysis, but that's kind of consistent with what we expected. And then the kind of hope of this method is that we're actually able to refine signals by modeling the pleiotropy, as we saw in the NCAN example. Future simulations, as I mentioned, we're going to simulate data using effect size and be estimated from real data. We've already started that. And then for this future real data, we're hoping to include a few more complex traits. And there actually are some methods now that are specifically designed to run MR in just a cis region or for a drug target. So we are going to compare against those, both in the simulations and the applied analysis. I'd like to thank all the supervisors that worked with this project, Mike, G, William, and Ewan. And thank you all for listening. And I'm happy to take any questions.
Great talk.
Thank you.
Great job, talk.
Thank you.
I think you need to work bacon into your example somehow. I think that would improve the talk.
Or what?
Bacon.
Bacon.
Yeah, that would improve the talk. No, seriously, are there questions from back here? Right, I was kind of joking about bacon, but I was thinking that's an example of the horizontal pleo,
Pleiotropy, people say. I always say pleiotropy.
right? Because there's more than one way to get to high cholesterol, right? There's the clearance of it in the blood,
Right.
and there's something telling the brain it has to have bacon.
Right. Yeah. Cholesterol can maybe be a difficult trait to use. I understand. There's multiple.
You can eat a lot of natural animal fat without getting high cholesterol. I'm an example of that.
Yeah, so it could be some
primarily eat fatty foods. I don't eat much and I stay lean. Most of the people that I know that follow my diet are that way. So, but anyway, I had some other follow-up stuff. So bacon is what you were gonna, because of the what were your
Talk is better with bacon.
yeah, everything is better. Yeah. So are you gonna make an R package for this?
Yeah, I have an R package right now. It's not like polished, but it will be more polished.
It sounds like this is really something that could shine some light on some dark areas that are.
Yeah, we're excited. Also, like I didn't talk about this kind of really similar thing called transcriptome-wide association studies, or TWAS or PWAS. They're kind of similar to our method as well, where they kind of use all the variants in a region, kind of do some shrinkage, and then you're basically doing MR, but you're not making any assumptions. So people kind of like accept in that case that you're going to get a ton of false positives. But really, our method is like really similar to that. We're going to do the exact same thing, but we can avoid a lot of those false positives.
Yeah.
So hopefully we can kind of convince people to try to do something like this instead of the traditional TWAS, PWAS, and even the MR stuff. So yeah, we're hopeful.
So can your method be extended using summary level data for the chain or something like that?
Yeah, so this is all, I just like, I didn't do any of that. This is all this is done using summary level data.
Okay.
There's two ways you can do that. There's kind of two ways, and I didn't see all the transitions. Yeah, basically when you're doing this variational inference procedure, you're doing like some coordinate ascent, basically.
Yeah.
And then each of the updates you can write in terms of sufficient statistics that can be recovered from summary statistics. Another way you can do this is actually you can just model the z-scores. You don't actually need to model X and Y. And so when we account for sample overlap, we actually model the z-scores, but it gives you the same thing. It's kind of two ways that you can use summary level data. Yeah.
All right, I think there's pizza. I'm gonna turn off the recording.
Yes.
Thank you very much. That was a wonderful talk.
Yeah. And I guess the other thing is if you can figure out a way to save people money, that's the way.